Now let's see the topic fate of ammonia. So fate of ammonia ante ammonia ఏమవుతుంది. So at physiological pH low. So let's see what is physiological. Usually it will be slightly alkaline, 7.35 to 7.45. So ammonia is protonated to form ammonium ions, means to the ammonia NH3 plus H plus ions gives rise to NH4 plus. So while most of the plants can assimilate nitrate as well as ammonium ions, so that will be taking nitrate as well as ammonium ions. So previously NH3 plus 2NH3 plus 2O2 gives rise to 2NO2 minus plus 2H plus plus 2H2O. ఇంకా అలానే 2NO2 minus plus O2 gives rise to 2NO3 minus. Okay, this is, we call it as nitrate. The nitrate and ammonium ions so are being easily absorbed, can assimilated by the plants. Later the quite toxic, so ammonium ions are quite toxic to the plants. So when compared to the ammonium ions NH4 plus and NO3 minus, ammonium ions are more toxic, hence we cannot accumulate in them. So let us now see how these ammonium ions are used to synthesize the amino acids in plants, because mainly ammonium ions are used for the synthesizing the ammonium ions, amino acids. There are two main ways for this. One is reductive amination. In this process ammonia will react with alpha ketoglutaric acid. So alpha ketoglutaric acid plus ammonium ions plus NADPH to form glutamate and NADP. So in this process ammonia reacts with alpha ketoglutaric acid and forms glutamic acid as indicated in the equation below. Okay, so they have given glutamate, so glutamate dehydrogenase. Okay, alpha ketoglutaric acid plus NH4 plus plus NADPH plus gives rise to glutamate dehydrogenase, gives rise to glutamate plus water plus NADP. What is transamination? It involves transfer of amino group from one amino acid to keto group of keto acid. So we are transferring the amino group from one amino acid to the keto group of keto acid. Glutamic acid is the main amino acid from which NH2 is transferred. The amino group takes place and other amino acid is formed through transamination. Means from one amino group, from one amino acid, amine group is being transferred to other amino acid. Like that, the transamination. So the enzyme transaminase catalyzes all such reactions. For example, here if you see, this is amino donor, this is amino acceptor. So now this is R1, this is R2. So from this we are transferring from here to here. So the two most important amides, asparagine and glutamine, found in the plants are structural part of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid. So respectively, by adding another amino group to each, the hydroxyl part of amino acid is replaced by NH2 radical. So the OH group is actually replaced by NH2 radical. So the hydroxyl part of the amino acid is replaced by NH2 radical, since amides contain more nitrogen than that of amino acid. They are transported to other parts of the plants via xylem vessels. So in addition, along with the transpiration stream, the nodules of some plants like soybean exports the fixed nitrogen as urides. These compounds are also have a particularly high nitrogen to carbon ratio. So if you see, the normally asparagine and glutamine are the amides. So what is the difference between amino acids, amide and urites. So amino acid is carbon, hydrogen. So one side you have NH2, one side you have COOH, and one side you have R group. So in this OH place you have NH2. So more amine groups will be carried by amides. So asparagine and glutamine. Urites also will have more nitrogen ratio than carbon. So this is what the fate of amino acid.